All right. Thank you again, everybody, for joining. We'll be expecting a couple more people to come in, I believe, over the next couple of minutes. Before we start and before I introduce our speaker for today, I would like to first just welcome everybody to our first Science of All Protein APAC edition seminar of 2024. We're really excited to kickstart this series here. And we will also have some upcoming webinars as well. So firstly, we have on April 18th, GFI APAC will be hosting a policy-focused webinar on the topic of decoding recent cultivated meat regulatory process in APAC. It will be hosted by our very own Ankur Chowdhury from GFI APAC. The webinar will be a panel discussion with speakers from Thyssen Biofarm, Majelis Ugama Islam Singapura, also known as MUIS, and the Food Standards Australia New Zealand Organization. We will be covering regulatory developments in South Korea, Singapore, and Australia, and what they mean in the cultivated meat industry. And yeah, we will link the Zoom link, the Zoom registration link in the chat now. Another upcoming news we have from GFI is that our research grant call is now live and accepting proposals. The funding is available for all protein research, and the proposals should target one of our 3 priority topic areas, which is upcycle plant proteins, next-gen fermentation downstream processing, and optimization of hydrolysate production to enable lower cost media. The deadline for this application will be 23rd May, and the link will be posted in the chat as well. But yeah, back to our event for today. Firstly, my name is Arun Naidu. I'm the SciTech Engagement Specialist at GFI APAC, and I'll be your host. It gives me great pleasure to welcome Dr. Danai Georgiadou as our distinguished speaker. Today, she talks to us about the species-specific strategies, tailoring approaches for in vitro growth of primary cell lines in cultivated meat production. Dr. Danai is a stem cell biologist with a strong background in primary cell line isolation and characterization. She's from the A-STAR Bioprocessing Technology Institute, also known as BTI. Informed by her work with diverse species ranging from crustaceans to pigs, Dr. Danai will delve into some of the strategies and methods employed to refine the isolation and expansion of novel cell lines. The talk will also cover the key factors that enhance the fitness and performance of the developed primary cell lines. Dr. Danai is currently leading the cell line workstream within BTI's CRISP Meats project, a major cultivated meat research initiative involving multiple institutions. Before we begin the talk, the talk will be for 1 hour, but the last 15 minutes will be for Q&A. On the bottom of the screen, you will see the chat and Q&A button. We encourage you to use the chat function to introduce yourselves to other participants, and also use this to direct your questions to Dr. Danai for the Q&A section. Once the Q&A section starts, I will go through the questions with the most upvotes and address that to our speaker. But yeah, that's it for me today. And I'd like to hand it over to Dr. Danai to begin our talk. Thank you very much.
Thank you, Erin, for the nice introduction. And of course, thank you at GFI APAC for inviting me today to talk a little bit about our work. I'm so excited to kickstart this series. Okay, let me share screen with you guys. Can you see the slides?
Yes.
Okay, perfect. Okay, great. So so today I will focus on our approaches in troubleshooting during our work on establishing a robust primary cell lines for cultivating meat production. So I hope you guys find tips and ways to actually face the bottlenecks that is very common for you to face in during your work with primary cell lines, especially while you were trying to develop like novel cell lines. Okay, so to give you an idea of what I'm going to talk about, I'm going to say some things about the background of our lab and what is this Chris Meets project is about. I'm going to talk about our work stream for the cell isolation, characterization, and expansion of the cell lines derived from terrestrial species, but also crustacean species that we work with. And also, I'm going to talk a bit about the benefits of adopting a species-specific approach towards your troubleshooting. And of course, where we are right now, where we are currently, and what are the future steps that we're planning to follow. Okay, so I'm quite confident that everyone that's in the field, and I hope everyone else that is not in the field, is familiar with the rising carbon footprint associated with agri-food systems. So I extracted this data not like a couple of days ago from the Food and Agriculture Organization of the United Nations website. So it actually represents how the CO2 footprint increases from 1990 to 2021. We can actually see like a huge increase after 2001. And something that is really actually interesting, if you notice, that there was a sudden drop in 2019. And it came back again in 2021. So I'm sure all of you know what happened during these years. So actually, basically, the humanity got locked down. So it's actually very obvious that the most sustainable way, the most efficient way to bring these upward trends into a plateau or even to increase it is to go into another pandemic. But that's not something feasible, and that's not something that we actually want to have. So in order for us to deal with these problems, we need to have other sustainable solutions. So the alternative proteins that are currently known. So it's first of all, it's like a fermentation, which is one of the most ancient technique actually. So and in order for it, how we take advantage of it is that the fermentation process combined with the power of the synthetic biology, we are able to produce proteins like animal proteins without the involvement of actual animals. And of course, the plant-based products that they're actually leveraging on the nutritional value of plants. And the new kid on the block that's actually still debating if it's a part of alternative proteins or has a new category of its own is, of course, the cultivated meats, which is actually the effort to create the actual food by taking biopsies, like a small amount of cells from the animal, and proliferating and replicating these cells in the lab. Okay, so so before I go into the more specific work that we do, I would like first to acknowledge and to acknowledge my team, my cultivated team. Actually, nothing would be possible without them, for sure. So basically, we're like a small team, and it's like a sub team of a bigger group that's called the Immune Cell Manufacturing Group that is led by Andy. So the other teams are Dr. Alison Lee and my partners in crime, Dorothy Chan and Yap Yongchi. And actually, Dorothy is the person that sees most of the work that I'm going to talk about has produced. And Yongchi actually is our creative genius, and I'm going to use some of her figures that she designed for the review that we published recently. So we are this small team. We are a part of a bigger ecosystem that is called Chris Meets. Let me hide this. All right. It's called Chris Meets. So actually, this project is it's involved many institutions. Like okay, so A-STAR is the host institution of Chris Meets, but also there are groups involved that they are from the National University of Singapore and the Singapore Institution of Technology, SIT. So these actually projects, we have many teams that we are specialized at every team specialized at a different part of the cultivated meat process. So you have groups that I am involved also in that do the cell isolation, differentiation, and characterization. Okay, so the brain master cell banking, which actually they take these primary cell lines and they verify the identity and the safety of the cell lines. Of course, groups for media development and groups that are specialized on creating the scaffolds, which actually the matrices that the cells can be grown onto. So it has to be edible and cost effective. And of course, last but not least, is the people that they actually create the bioreactors, which they're going to give us the opportunity to upscale the production of cultivated meat. So actually, Chris Meets, the goal is to set the foundation of the cultivated meat research, and in order for us to be able to address problems and obstacles and bottlenecks that startups, cultivated meat startups, and biotech MNCs are facing, in order for all of us to make Singapore a manufacturing hub for cultivated meat products. Okay, so going into the actually what is the process of the cultivated meat production, I'm gonna it's a very simplified process, but it stands for every type of species that we're working with. So we start with the collection of the specimens, which is the animals, either it can be seafood or like you hear, all meats or mammals. And then we perform tissue biopsy and dissociation, where we're isolating the cells. And then we try to expand them in vitro by optimizing their media, the growth factors, and the supplements that the main needs. And that is in order for us to be able to create a big cell biobank that is going to be our foundation for further cultivation meat development. And so these cells, after they're optimized and becoming a robust cell line, they're going to be used they're going to be expanded on matrices, on scaffolds, that we call them. And these systems, they're going to be upscaled into a huge bioreactor in order to increase the biomass of these cells. And then the final product, of course, is the harvesting of the cells and the processing into the final product. So my team is actually focusing on the first part of this process. So we actually are responsible to locate the stem cells from the tissues that we are working with and optimize them, making them robust cell line, and then contribute to the expansion of the cell biobank. Okay, so the technology behind this is being developed for many years, especially for regenerative medicine and biomedical research in general. So to locate the stem cells and to make them to make them proliferate outside of the body. So two main sources of the stem cells are embryos and juveniles and adults, so somatic cells. So from embryos, the cells that we're getting are embryonic stem cells, which they have the pluripotent cells, and they have the capacity to differentiate it to the different type of cells. And from the somatic cells, the juveniles and adults, you can isolate either terminally differentiated cells or adult stem cells, which is mesenchymal stem cells and precursors cells like the muscle precursor cells. So in the case of the terminally differentiated one, because they cannot actually differentiate the type of the cell that you want, we need to direct reprogram it into induced pluripotent stem cells. And then when you have the stem cells that you need, they're going to be self-renewal, differentiated in the type that you are interested in. And in the case of cultivated meat, we're gonna go for muscle, fat, and connective tissues. But in the medicine, in the medical research, actually, you can differentiate it in any type of tissue that you would like. So the thing is that the tools for this big process are optimized and developed mainly on the human species, for the human species, and actually and all the antibodies and the small tools that you need to successfully differentiate, successfully produce the serotype that you want, is either mammal genome, or a model that actually can be translated for human pathophysiology and physiology. So and all the species actually that we use for research, definitely we are not interested in eating. So that gives us a little bit more troubleshooting that we have to do in cultivated meat, because the whole process, right? In order for you to find the stem cells, you have to locate this as a stem cell niche in the tissue that you're working with, and then also to find enzymes and buffers that actually are gentle for the cell but are hard for the tissue, in order for you to isolate the single cells. And then, of course, then expansion and characterization. You need validated biomarkers, antibodies that actually work against your target and give you signal, and optimized omics tools. And that it can be even like as simple RNA and DNA extraction kits. It may not work with the species that you're working with. So, and of course, the evaluation of the differentiation potency, which also needs extra antibodies for specific markers for the specific genes, and other tools which has like the similar issues. But the thing is that if the genome of the species that you are working with is annotated, then it makes the things a little bit more straightforward, because you can actually work towards a trans-species translation, meaning that you have the markers, the biomarkers that you're looking for to characterize your cell lines in human or mouse species. And then by comparing the genome that you're working with and the genome that these markers are validated, and actually evaluate the percentage of the conserved regions, you can actually use these markers to further validate your cell lines from different species. And that is mostly possible for terrestrial species. So I put some representative images here from our chicken cell lines. So we work with two breeds of chicken. One is a kampong chicken, or else the village chicken, which is quite common here in Singapore. And also black chicken, which originated from Indonesia. And actually it's very famous for its hyperpigmentation. And actually a fun fact about this is that even the cells are black. So it was quite surprising to see when I first started working with them. So you see on the top left, we have the muscle stem cells that we actually isolated from a biopsy that we received from the breast of the animal, and the antibody-derived stem cells we isolated from fat pads that are located in the abdominal part of the animal. Okay, so the next step after the isolation actually is to validate that actually the cells that you have under your care is the type that you want. So in order to do that, there are like very common markers that have been validated a lot in the regenerative medicine and biomedical research, which is PAX7, myogenic factor 5, PAX3, MyD1, MyoG for muscle stem cells. And these markers actually identify different stages during the differentiation from the stem cell to the terminally differentiated gene. So in order for you to have an idea, the types of population that you have in your primary cell line. And for the adipose derived stem cells, you can use the most common one that is being used is PPAR gamma, which is an early marker of adipogenesis. And then in order to validate your cell lines, you just evaluate the expression of these genes in your cell population. So personally, I prefer for mast stem cells to be validated with quantitative qPCR, so with evaluating the expression of the genes inside my cell population. And for the ADSCs, which is adipose derived, I prefer to do another technique, which is a flow cytometry, which is actually actually taking like a positive or negative signal for surface antigens, which actually basically is the same. It's like markers that they are specifically expressed only in the cell of interest. So here you see another species that we're working with, which is porcine. That means pig. And at the top you see the mast stem cells, and below the adipose-derived stem cells that were isolated from the subcutaneous fat, which is like under the skin of the animal. And in order to validate the ADSCs, we chose markers, the two on the top, the markers that are specific for mesenchymal type of stem cells and ADSCs. And we check the stemness of the population by targeting for CD90, and of course the negative control in order to exclude any other type of cells in our population that we actually don't need. Okay, so with the terrestrial species, you can see that it's a little bit more straightforward. There is some troubleshooting, of course, because humans are different from animals. But it's something that you're eventually gonna get there. But with aquatic species, it's a different story. So actually, the crustacean species, it was the first species I worked with when I got involved in the cultivated meat field. It's still my favorite because it's quite challenging. So I'm still working with tiger prawn. So when we actually received the animal, there was a lot of many different rounds of optimization and validation, and we try to locate the stem cell niche for different types of cells. And after many rounds, we successfully isolated muscle-derived cells that you can actually tell that they have the phenotype of the satellite cells, which is actually the mast stem cells when they're in the quiescent state. And so they're like they are preferred to be preserved in suspension culture, so that adhesive capabilities are very low. And they're in the specific for the prawn, the size is quite small. It's 4 to 5 microns. And also, we were able to isolate cells from the fat pads of the tiger prawn. And again, we found cells that they have the characteristic of mesenchymal type of cells, which is the ADSCs, which is adipose-derived stem cells, which is quite adherent to the surface of your culture, and they have spindle-like morphology. So the thing is that, okay, so we have this, we managed to have cells to work with, and now we had to actually validate these cells to see what we see actually what we want. But in the case of the crustaceans, and I'm sure for a lot of aquatic species, there's no validated biomarkers, there's no available antibodies, and of course, there's the biggest challenge is that the genome is not completely annotated. So we say, okay, so we have to stay to go like a step back with our process, and use the genetic material that we can extract from these cells in order for us to do some sequencing and then be able to actually identify targets that can be used for the validation. But then it was another problem. So even if we managed to isolate the cells, and actually with a quite optimized protocol with high yields, these cells, they were barely moving. They weren't proliferating. And the surprising thing, the interesting thing is actually that it's not that they were dying, they were apoptotic. They were just staying in an idle state. So so we came into the chicken or the egg paradox, which actually we don't have an annotated genome, and then we don't have a sufficient material in order for us to be able to sequence and identify like possible targets that we can use. And also there's no working antibodies, and the biomarkers that can actually identify, they haven't been validated. So we decided that it needs to redefine our approach and go a step back even further. So we said that, okay, this approach should be not trans-species translation, but it's going to be species specific, completely species specific. So that means that we went back to the troubleshooting, and we optimize the growth condition and the media composition. And in order to do that, I mean, if you think about the first growth media that they were used in the 19th centuries, it was actually tissues that were growing in the serum and the plasma and of the animals, right? So with this in mind, we were based. We wanted to actually to have a list of different compounds to test with our cells. Our cells, they're based on the composition of the hemolymph, which is the equivalent of plasma, but in crustaceans. So we got, so we tested multiple compounds actually that they have been found in the hemolymph of the tiger prawn. And either we screen them in there alone or in the combination with each other. And indeed, actually, we see that that there were combinations with the media type C, that it decreased, it initiated the proliferation of the cells. So unfortunately, I cannot disclose the specific of these these formulations, but because we still be writing now the publication, so it's going to be available for everyone. But the thing is that it wasn't enough. It's still not enough. The cells are still not fit enough to be processed further for the cultivated meat. So we decided to go a step further, right? So apart from the media development, we wanted actually to look more on the abiotic factors. So that means we all know about the pH, the temperature, and osmolality, right? And especially the osmolality for aquatic organisms is really, really important. And I cannot stress it enough. And not only for the growth media, but also for the every different buffers and every different stages of your process. But apart from that, we wanted to take something different. We want actually to see what's the impact of pressure and oxygen on our cell lines, so the crustacean cell lines. Because don't forget that crustaceans are actually deep sea creatures, and they thrive in an environment that we cannot even survive for seconds. And they survive in high pressures and no oxygen. So we are really setting up now a process. We can actually gonna study and manipulate the pressure and oxygen conditions of on the cell culture to see if actually we can increase their metabolic activity. Because if you think about it, higher pressure it creates the chemical reactions of the body to increase in speed, and the metabolic rate is changing. And on this note, actually, it's not that the first time that when we try to replicate the microenvironment of the cells, it's not the first time that that researchers showed the benefits to be more species specific, to be more environment specific, microenvironment specific. So you can see if you go into the biomedical research, there are like many groups that they work by do hypoxic conditions. So that means like very low oxygen, and see the effect on the mesenchymal stem cells. And you can see that actually, because we all know that, okay, you culture the cells in vitro, but inside our body, between our tissues, we don't have 20% of oxygen. We have maximum like 2% of oxygen. So you can actually see that when you put the cells in an environment that actually they were programmed to develop in, they have a completely different reaction. So it's also the same with the hyperbaric, which is the high pressure. So you see there's especially with stem cells and neurons, the effect is quite promising. Okay, so our next steps is actually with a collaboration with another group to create a 3D printed dust chamber that we're gonna use for our hyperbaric and hypoxic hyperbaric experiments, not only for the crustaceans, but also with all the cell lines that we have, and possibly address like the most challenging bottlenecks in the primary cell line development research, which is the suboptimal growth rate, because we need a specific growth rate for the cell line to be qualified as a proper cell line for cultivated meat purposes, the stress phenotypes, which is the cytosolic vacuoles and the expansion of the cytosol, and of course apoptosis, and the premature senescence. And also something really important, the stemness loss, because if the stem cells spontaneously differentiate, we can actually lose our whole line. Okay, so to summarize, the trans-species translation approach may not always be optimal for species that are very distant on the phylogenetic tree. So you can see the mammals and the crustaceans are the furthest apart in evolution. So that means that that a lot of the like a very small part of their genome is conserved between them. So introducing species-specific combination of nutrients, hormones, and growth factors. So the media development has shown like benefits for it. So but the thing is that this is not enough. That the media development is not enough, and more other factors like the biotic factors should be optimized also, especially for aquatic organisms. Hopefully I'm gonna have soon results for you guys, and for the next time that I'm gonna present the work that can help with the troubleshooting and the bottlenecks that we are all facing with the primary cell lines. And last but not least, I would like to, apart from our culture mates, I would like to thank all my colleagues under the Immunocell Manufacturing Group for their support and their in these troubleshooting ventures of mine. And also, of course, our research partner in the CRISMITs. It's other groups under A-STAR, other institutes, also the NUS and SIT. And of course, the funding source of CRISMITs, which is the SFA and A-STAR and the NRF. Okay, this is what I want to talk to you about. And because CRISMIT is all about collaborations, feel free to contact me in my email and for further discuss any issues that you may be facing. Thank you so much.
Hello. Yeah, thank you so much for the great talk, Dr. Danai. I think that was really insightful. I definitely have some questions, and we have I think 2 questions in the Q&A box right now. But to all of our other attendees, yeah, please, whatever questions you have, please add it in here, and we'll start going through them. Yeah. So yeah, firstly, we have a question asking, how did you realize that the muscle-derived tiger prawn cells were suspension instead of dead adherent cells that floated off? So yeah.
All right, so it's very easy to distinguish actually suspension cells from dead cells because of the membrane is not very intact. So the chains shape they don't remain round in a period of time. And also we see they're idle, but they still proliferate like really, really slowly, but they do. So and actually we also have we also have we know the apoptotic phenotype, because it's not that all the cells that we isolated were viable. So we can actually, it's really easy to distinguish between the two states of the cells.
Okay, yeah, that makes sense. And I a little bit on that too. One of the questions I had is, could you explain a little bit more why are different techniques needed to characterize markers from maybe the chicken cells versus the porcine cells? I think you mentioned qPCR and flow cytometry. So my understanding is that the markers are different in itself, as you listed, but I'm just curious, what is it about these specific techniques which make it easier for each of the cell types?
Okay, so the thing is that with the markers, the easier that the more robust, the more high in expression that the markers are in your cells, the easier it is for you to validate your cell line with not having to have a big yield of DNA, or when you are like limited in the quantity of the DNA that you are working with. I personally prefer the mast stem cells that there's a whole list of different genes to actually to be able to use. It's easy and straightforward to do like a quick qPCR and validate your cell line, having the appropriate controls. And on the other hand, with mesenchymal stem cells, which is the adipose-derived stem cells, the most robust way to do it is to identify the antigens on the surface of the mesenchymal cells, and it's quite established and optimized for human and mouse, and this, especially with mammals, like the region of the antigen that is being targeted is quite conserved among mammals, so it's easy for you to validate quickly and effectively your cell line.
I see. Okay. Yeah, thank you for addressing that. Our next question from our audience is, have you ever worked with fish cell lines? Would it be the same challenges as tiger prawns? In the case of tiger prawns, how long does it take to get the muscle and adipose-derived cells?
Yeah, so I work with fish cell lines. I haven't developed. I haven't had the. I haven't done the isolation from the fish tissue, but I work with cell lines, with fish cell lines. So there's a difference in the phenotype between the two aquatic organisms, and actually you can see that that the fish with our vertebrates are more involved, that are further down in evolution than the crustaceans with our invertebrates. And you can see that the fish cell lines are more plastic in adapting in the environments, but also you can see that it's the way that they behave and the growth rate that it's quite low is not optimal. So I still believe that by changing the abiotic factors, which like the pressure of the oxygens that these cells are going under, we can actually achieve higher metabolic activity for the fish cell lines. So for the crustaceans, how long is it depends on, you have to find the right spot in the tissue. So the thing is that it took us a while to identify where the stem cell niche is, but after you do it, after you know where to look at, then it's a one-day process.
And a little bit on top of that too, are there any reasons for the crustacean cell lines that there are no validated biomarkers, no available antibodies, or no annotated genomes? Is there a reason why maybe that work has not been done or prioritized yet by the industry?
Sure. Okay, so of course it's because it wasn't the species of interest to begin with, but also the aquatic agriculture. Let's say there is a lot of research on prawns, but the fact that since the 1960s, where they started actually trying to develop the in vitro cell line, there's not a single cell line developed that actually can be used for further studies. So it goes back into the whole paradox of the chicken and the egg. So if you don't have the material, you can actually don't develop the markers that you need in order for you to identify the cells. So it's because of that. So it's the same reasons.
I see. That makes sense. Our next question from the audience is, can you share any high-level details about what made Media C unique compared to the other formulations? For example, what was it to do with optimizing certain lipids, amino acids, or any other type of macromolecules?
Sure, I can give a hint about it. If what makes special this type C is actually that we included specific types of hormones that they are present in a specific period of the life of the prawn. So okay, so to explain that better, crustaceans are seasonal. So you can see that you're getting like more robust and better results with your cell lines when actually you isolate cells from their breeding and the molting period. So by actually identifying like compounds that they are important for this actually process, you can actually help a lot your cell line in vitro.
And I guess the other details we'll see in the publication.
Yeah.
I think we'll all be looking forward to reading that as well. Our next question from the audience that we have is, thank you for the wonderful presentation. Are you able to comment about sample isolation related contamination, and how do you tackle this with your everyday work? So maybe some examples could be good.
Sure. Okay, so everything in cell culture is about having like sterile conditions, right? So this comes with practice. But okay, with the crustaceans, with aquatic organisms, and basically for crustaceans, it's a little bit more challenging because they don't have innate like immune system. So actually, they are defending diseases every time that they come. So they don't have a memory in order for them to they go or live basically with protozoa, with many different bacteria. So it's a little bit more challenging, even if you actually work under sterile conditions, to eliminate all these contaminants from your process. So the best thing that you can do is actually optimize the sterilizing process as soon as you get the tissue. It's that you can actually try different solutions, either like povidone, it can help, or bleach, or ethanol. So it's actually to see how much the tissue can survive while you are processing it. So it's like trial and error. And the thing is that if you actually eliminate as much as possible from the tissue biopsy states, then you can see that your cells have like a less chance to get contaminated later on, to increase bacteria.
And maybe just for our audience to know too, like within your everyday work, like how often do you or your team run into contamination issues? I understand that you still have to like optimize the process, different cell lines, but yeah, how often is this an occurrence for you?
Okay, so for the terrestrial species, we are lucky enough not to have contaminations without even antibiotics in our cultures. But of course, it's going to be a random contamination at some point. We're humans. But with crustaceans, after we optimize this process, yes, let's say like if we do like 5 isolations, one of them is going to be contaminated. So it's going to be something like that. Yeah, so the things that you more, at the beginning, I had way more contaminations, especially with my aquatic cell lines. So you see that when you're working on the sterilization part, then it's going to help a lot your cells, but not overdo it because your cells may die. So it has to be like a balance.
I see. Okay, because our next question is actually, have you found shrimp cells to be particularly challenging to work with when it comes to contamination? I've heard that from others, and wondering how general of an issue it is. So yeah, I guess that question addresses it. Your answer just now addresses that as well.
And also something that we are exploring actually is that we're gonna add antimicrobial peptides. So this is like something like an idea that it's developing by other groups. And maybe if you guys have like some peptides, the microbial peptides that you can actually put in your culture, just do it. Just try and see.
And maybe a little bit on top of that, a question I had is with the 3D printed chamber, which you'll be developing. Like, are there any types of like any materials you're thinking of which maybe would be more optimal for the crustacean cell lines, or maybe to also avoid issues around contamination? But as you're designing what that chamber looks like, but yeah.
So I'm not going to be the one that's designing. So the thing is that
Okay.
we talked like with the specialist people that they're going to actually take care of it. So we haven't started yet. So I don't know. Probably we're going to start like evaluating different materials and see what problems we're facing, and then move forward with this.
Okay, yeah, that makes sense. Our next question is, do you face any challenges with impurities from other cell types when isolating muscle stem cells or adipose-derived stem cells? If yes, how do you deal with this issue?
Sure. Okay, primary cell lines, like it's a mixed population of cells, right? So with the muscle stem cells, you have to find the best. For me, what works best is to play around with the speed of a centrifugation in order to separate the satellites, the myoblasts that are very low in molecular weight, so they can stay in your suspension, and the bigger cells, which are going to be the immune cells, fibroblasts, and all the other type cells, they go down, so you can actually separate them. And that between species can differ a lot. So you get an idea basically of how big is the organism and how big are the cells are, in order for you to start playing around in a range of the centrifugation speeds. But this is how you tackle with this, and and that's not like 100% purification of the cells, but later on with replating, because it's the myoblasts, they stay there, they have really low adhesion capability. So with replating, then you get rid of the other type of cells.
Mm-hmm.
And of course, then you characterize it to see if the population is pure, or you do. And if you see that there's like a big issue of mixed population, maybe another idea is to do the single cell cloning and then create your cell line from a single cell. But that can be challenging sometimes. So, but it's possible. It's possible to do. And with adipose-derived stem cells, the good thing about them is that they love to attach on surfaces. So if like after 24 hours after your isolation, you can actually wash your well, and you can see that everything that stays on is the cells that you move. So this is with the ADCs is quite actually it's quite easier.
I see. And in terms of the purity level, what sort of threshold are you going for in this, like for it to be a viable experiment?
Okay, so the thing is that the more passages that you go with your cells, the more pure they become. Because if you think about it, the terminally differentiated cells, they die off at some point, because this is the difference between the stem cells and terminally differentiated cells if they're not spontaneously immortalized. So for us, if we have like 80-90% of purity in our cell population, it's good enough to move forward, because it's actually the majority of the group that is in the population.
I see. And just one more question on the purification. So centrifuge, I guess there is like a cost and ease aspect to it as well. Are there other maybe like high costing, more maybe more precise forms of like purification techniques which could be used, but maybe you're not really using from like from maybe a costing perspective?
Sure. I mean, they're like either the FACS, right? Flow cytometry that you use. You can actually sort the cells based on the surface markers that they have. But that's you have to have an antibody that works. You have a validated antibody, because it may give you signal, but maybe it's not the signal that you want in the different space that you use. So that is actually something that it will come when specific antibodies will be developed, but it's also more costly.
I see. Okay, and then yeah, we have I think three more maybe yeah, just two or three more questions from our audience. Our next one is what are the estimated costs associated with quite connected to what I was asking, but what are the estimated costs associated with cell-based production process for the tiger prawn project?
Okay, so the thing is that because it's quite like the state that we are right now, it's still quite primary. We are just try out to make the cells, to wake up the cells, right, to grow them. So now the costs, we try to keep it low, but it's not a factor that we take into consideration at this stage, because when we actually learn more about their metabolic activity and their physiology, then it's going to be easier for us to optimize it and make it like lower cost. But first, we have to find out what's going on.
Okay. So still maybe a whiles away from optimizing for cost. So
Yeah. I mean, the
yeah.
cost is an issue in the cultivated meat industry, right? So crustaceans are going to be the last players to to join.
The next question, maybe next question, two questions is a little bit not just specific to crustaceans, but more general to cultivated meat. Is firstly, do you have any recommendations for guidelines regarding the establishment of a cell bank for cultivated meat production? So this seems like maybe tips and advice for people looking to start like a cell bank. Yeah.
Sure. Okay, so first, like the biopsy that you actually gonna select to isolate the cells from, you have to have like documentations that the animal is healthy. So don't go all the way into generating your cell line and then you're gonna use it because of the regulations. So that is the first step. And another characteristic of the cell line, in order for you to be able to say that it's ready for further processes, is that they have proliferate a growth rate close to 24 hours, and that it can actually grow more than 20 passages, depending on the subculture dilution, of course, but that they survive more than 20 passages, that can actually say that this line can be used for to optimize other stages of the cultivated meat production and actually be the foundation block of the final product.
Yes.
And
Yeah,
of
sure.
course, contamination free.
Yeah. Yeah, super great to be able to share these insights with our audience as well. Our next question is the audience is asking, would basic conventional meat taste and texture is quite related to aging of the particular animal? Is there a way that in cell-based meat, can we ensure or vary this, maybe based on its proliferation is what this member is asking, to get different types of taste and texture, which would maybe correspond with like the age of the animal.
Sure. Yeah, so I don't know if it's more about the age of the animal or like the post-modern modification after the animal is dead, right? So you take like the steak. So there is of course some metabolic activity happening, something still changing, and actually this is how we call it the meat is aging, right? Because it's actually there is some processes that are happening. So there is research on it happening right now, actually, how the umami taste or the other type of tastes are going to be affected in comparison with conventional meat, with cultivated meats. Because also, if you think about it, the cells in vitro, they're not sort of contract that they do in the animal, and that can this energy release, like difference between the two cell types, it may affect also like purines are being like produced, and with purines are like related with with different types of tastes. So it's something that it needs to be further studied. And
I see. And no, yeah, thanks for addressing that. Yeah, quite a unique question, which yeah, maybe I didn't think of earlier as well. And yeah, towards the next question, maybe I just wanted to point out one of our team members put in the chat about your column, which you wrote for GFI APAC, about outlining some of the important abiotic and biotic factors to consider for crustacean cell line development. So everybody in the audience, yeah, Manasa has put that in the chat, so I would recommend people click through to read that as well. But the question I had is, you mentioned pressure and oxygen as abiotic factors to experiment with for crustacean cells, and even showcase a lot of the work being done. What are some other abiotic factors which are varied to maybe try to optimize for porcine or chicken cells, or even, yeah, maybe beef cells, just I guess outside of crustaceans? Yeah.
Okay, of course, it's the temperature, because every different animal has different body temperature, of course. And so this is a biotic factor that is quite important. And for me, I saw with the cell lines that we work with that also correcting the osmolality, that we take some things for standard when we do like the research that we don't even take with our the growth media that we're actually buying with DMEM and the other media, growth media that we actually use. So by adjusting the osmolality, your cells are going to behave way better. You're going to see them that there's going to be less stress. So by actually check if there's any difference in the numbers in the osmolality of the plasma of the animal, you can actually correct also this factor. And yeah, so with the mammals, I think, which is a little bit more straightforward, if you play around with your with the temperature and like the osmolality, as I said, of your media, it's gonna give you at least like a good foundation to further optimize.
I see. Yeah. Thank you. Yeah, thank you very much. So yeah, we have 2 minutes until 11. I want to be mindful of everybody's time, but I think we will close the Q&A as of now. But thank you so much for taking all of that time to give really thorough answers to all of these questions. I really do believe it's really insightful for an entire audience, and later this will be on YouTube as well. So I think for other people to see. And also really a big thank you from all of us and GFI APAC for a very insightful talk. I think it would be really great to see the progress of your research, your publication, and hopefully get you back here in the future to discuss all of the other things you have worked on. So yeah, thank you very much. And yeah, we'll close our session for today.
Okay, thank you so much for the invitation, guys. And I'm looking forward to come back and say some results on the on the abiotic factor like exploration.
Thank you, everybody. Have a lovely, lovely rest of your day. We'll close the meeting.